Hi, my name is Esteban. Me and my colleagues come from the Jaume I University in Spain, and I'm here to present you our latest research. I'm going to talk about the hemispheric lateralization of language and how its atypicality affects inhibitory control in the human brain. One of the oldest findings in human neuroscience is that some cognitive functions are hemispherically specialized. Back in the 19th century, Paul Broca described that language was functionally lateralized in the left hemisphere. However, modern neuroimaging techniques have allowed us to be more precise, specifically when looking at activation in the inferior frontal cortex or Broca's area. We have seen that language is typically lateralized on the left hemisphere in approximately 95% of right-handers. But when looking at left-handers, this number goes down significantly. Approximately 1 out of every 4 left-handers not presenting this typical left lateralization. These really rare atypically lateralized individuals present a distribution of language in either the right hemisphere or bilaterally controlled by both hemispheres. But language is not the only cognitive function known to be hemispherically specialized. So, what happens to these other lateralized functions when language is not typically lateralized to the left hemisphere? Well, there are two main hypotheses in this regard. The statistical hypothesis postulates that hemispheric lateralization of every function is defined independently of the others. Hence, a shift in language lateralization should not affect the lateralization of these other functions. On the other hand, the causal hypothesis postulates that the lateralization of every function are intertwined somewhere along our ontogenetic and phylogenetic development. So, a shift in language lateralization should be followed by a shift in the other functions. Although there are not that many studies that have tested these two hypotheses, current evidence mostly support the causal hypothesis. When we explore atypically lateralized individuals from language, most of them present a mirrored organization of their other functions. So, a person that presents language in the right hemisphere would have a mirrored representation of prosody, visuospatial attention, and face processing on their left hemisphere, while also showing praxis and word processing on their right hemisphere. But surprisingly, even though atypical language lateralization is mainly defined by looking at the inferior frontal cortex, no one has tested what happens to the function of its right homologous. And that is precisely the objective of our study. Okay, so to you all wondering what the right inferior cortex does, it primarily mediates inhibitory control. Inhibition can be defined as the suppression of a response tendency when you are presented with a cue, whether it is external or internal. A lot of neuroimaging evidence describes that inhibition originates as a right lateralized projection from the right inferior frontal cortex to the right subthalamic nucleus, with additional participation of the right parietal cortex and the right presupplementary motor area. So, in the present study, we used fMRI to explore what happens to this right lateralized inhibition network when language is atypically lateralized. So first, let's talk a bit about our methodology. We modeled inhibition using the stop signal task, which is the most used paradigm for this function. This is an event-related task consisting of both go and stop trials. In the go trials, participants must respond with a button pressed as fast and accurately as possible to a really simple lexical task. Is the presented letter stem a word or a pseudo word? However, in about 20% of trials, they will be presented with a stop trial. The stop trial is nearly identical, except that shortly after the presentation of the letter stem, participants will hear a beep sound. When they hear this beep, they have to stop their response and don't press any button. This stop condition will be our condition of interest. As for our sample, we preselected 86 individuals according to the hemispheric lateralization of language. 50 were typically lateralized, whereas 36 presented an atypical lateralization of language. So, we presented our participants the stop signal task during an fMRI session, and then we compute the functional asymmetry maps of their task. Functional asymmetry maps informs us about which voxels are significantly different from their hemispheric voxel counterpart, okay? Its calculation is fairly simple and starts with the original contrast map of each subject during the stop signal task. We create a copy of this image and then we flip it by midline, exchanging the position of both hemispheres. Finally, we voxel-wise compute the difference between the original and the flipped image. As a result, the value in each voxel now informs us about how different the brain activation is between both hemispheres. And here we have the results from comparing the functional asymmetry maps of the stop signal task between the typically and atypically lateralized groups. The significant clusters on this image are the brain regions showing asymmetry differences between both groups. Namely, the 6 regions are the inferior frontal cortex, the presupplementary motor area, the angular gyrus, the middle temporal gyrus, the thalamus extending into the subthalamic nucleus, and the caudate. These clusters correspond surprisingly well with previous descriptions of the inhibitory control network. For reference, here we have the mapping by Aaron and Poldrack that I presented to you earlier. Similarity, as you can see, is pretty impressive. But we still need to know the direction of these asymmetry differences. For this, we have to take a look at the following graph. This graph represents the activity of these 6 regions in both hemispheres. The bars extending from 0 towards the left represents left activity, whereas the bars extending from 0 to the right represents the right activity. Also, blue bars correspond to the typical groups, while orange bars correspond to the atypical groups. So we can see that in all 6 regions, the blue typical bars are longer on the right side, indicating a right lateralization of the inhibition function. However, when looking at the orange atypical bars, we find an opposite pattern. Activation is greater on the left hemisphere. Hence, in these atypical individuals, the whole inhibitory control network is mostly supported by the left hemisphere. We also calculated the laterality index for both language production and inhibitory control in the inferior frontal cortex. The laterality index is a proportion of the brain activation between both hemispheres in a particular contrast and ranges from -100, that will be a totally right lateralized function, to +100, that will be a totally left lateralized function. And as you can see, we found that the laterality indexes of both functions were inversely correlated with a coefficient of 0.58. That is, the more right the activation was during language, the more left the activation was during inhibition, and vice versa. Lastly, we also explored if atypicality was related to differences in performance during the stop signal task. So we explored how fast they responded and how fast they inhibited their responses. However, none of these measures showed significant differences between typical and atypical groups, whether by language or inhibition. So let's sum up our conclusions. First, our data here shows that inhibitory control shifts its lateralization in concordance with language. That is, atypically lateralized persons who process language using the right hemisphere present a mirror organization of its inhibition into the left hemisphere. Second, we found that this mirrored organization affected not only the inferior frontal cortex, but the whole inhibitory network, from cortical regions to subcortical nuclei. And lastly, we could not relate the typicality of lateralization to neither a worse or better performance. So, before ending this presentation, let me give you a brief insight to where is our research project heading up from here. If we look back at the correlation chart between language and inhibition, we can see that our participants can be classified into 2 different groups. Some individuals present a strong segregation of both functions. These individuals are located in the green squares and we call them segregated. The rest of our participants present a weak lateralization of either language, inhibition, or both. These individuals are confined in the red squares and we call them integrated. The analysis we are currently working on involve the comparison of these 2 groups. These comparisons informs us about the consequences of having a strong hemispheric specialization or not in a particular brain region. In our case, this region is the inferior frontal cortex. Importantly, I can tell you that our preliminary analysis using this approach have yielded really interesting results. For instance, we did find performance differences using this classification. Integrated individuals, that is, those with weak language lateralization in a function, were slower when inhibiting their response and also made more mistakes during a reading test. Differences in interhemispheric connectivity also existed between these individuals. We found that the integrative group had significantly more connected frontal lobes, both functionally and structurally. We think that these preliminary results would support the idea that hemispheric specialization evolved as a mechanism facilitating cognitive efficiency in the brain, and we are really eager to present them to you as soon as they are ready. So, before ending, let me thank my colleagues at the NNF and the UJI back at Spain, and to the Spanish Ministry of Economy for making this research possible. And of course, thank you all for your attention.